buffer systems of the blood. The buffer systems available in our blood. So in the background, you can see that the buffer systems, main two buffer systems, sodium bicarbonate buffer system, carbonic acid buffer system, and everything is there that is connected with the lungs as well as the kidney. So we'll see the buffer systems now. So before going to the buffer system, you must know what is buffer solution. That already we have seen in the previous one. So what is a buffer solution? A buffer solution is it is a solution which maintains its pH constant. Always it maintains its pH constant even upon the addition of small amount or drop of an acid or base or alkali. So there are two types of buffer solutions based on their pH. Acidic buffer, basic buffer. Acidic buffer will be having acidic pH. Basic buffer will be having pH more than 7. It's greater than 7. What is this acidic buffer? Acidic buffer is a combination of a weak acid and its corresponding salt. That is a weak acid together with a salt of the same acid with a strong base. They are called as acidic buffers. For example, already we have seen this one, acidic acid CH3COOH plus and its sodium corresponding salt, sodium acetate CH3COONA. If you combine these two, you will get an acidic buffer. Next one is basic buffer. It is a basic buffer is a weak base, combination of a weak base and its corresponding salt with a strong acid. They are called as basic buffers. And buffer systems of the blood. So what are all the buffer systems available in our blood? So buffer systems of the blood. Buffer systems of the blood. Why do we need a buffer system in our blood? What is the importance or significance of a buffer system in the blood? If the acids produced in our body, because during metabolic reactions, acids are produced in the body from the catabolism of the food material and other cellular processes, that is other biochemical reactions. Then these acids are not removed, or if they are not buffered, what will happen? The body's pH would drop down. That is acidic side. It will go acidic side. pH will come down to acidic side. So in order to avoid that one, the buffer systems are available in our blood. So significant drops in pH, even in 0.1 pH change, drop in pH interferes with the cellular enzymes and enzyme systems. So that is why pH of the blood should not be changed. So how to keep the pH of the blood constant? Using buffer systems. So there are 4 major buffer systems available in our blood. One is protein buffer system. That is with the help of the amino acids, protein buffer system is formed. Second one is hemoglobin buffer system. Third one is phosphate buffer system. Fourth one is bicarbonate carbonic acid buffer system. So 4 major buffer systems are available in our blood. So when you see that the maintenance of our blood pH is 7.4 neutral. So you have to maintain that blood pH is 7.4 always. So for the maintenance of the blood pH, it is very, 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 very important for the proper functioning of our body. And it can be critical if this pH of the blood is not maintained. So how to maintain the blood pH? Using buffer systems. So the main buffers are buffer systems in the blood that is going to maintain the blood pH are bicarbonate buffer system, hemoglobin buffer system, protein buffer system, that is plasma protein buffer system, and phosphate buffer system. Mainly the buffers which are involved in the buffer system are mainly carbonic acid. that is H2CO3 is carbonic acid. It is a weak acid and its corresponding salt is bicarbonate ion, HCO3 minus. This is a conjugate base. So this is going to act as a main buffer system in the blood. So H2CO3 weak acid, it reacts with water and you will get a conjugate base bicarbonate ion HCO3 minus plus H3O plus. So this forward and backward reaction is going to maintained at equilibrium state so that the pH of the blood is maintained. So this acts as a buffer system, bicarbonate buffer system. So we'll see this bicarbonate buffer system in detail. Here you can see that the pH of the blood is maintained at 7.4, but that is the neutral pH, that is the normal pH of our blood. So it is maintained at 7.4 by the carbonic acid bicarbonate ion buffer system. See, this is that buffer system. H2CO3, HCO3 minus, and acidic substance. When you add some acidic substance with this one, then what will happen? Blood will be moving this side. The pH of the blood will be moving this side. HCO3 minus and H2CO3 level changes. If alkaline or base substance is added, pH will be moving this side, alkaline side. So when an acid enters the bloodstream, what will happen? This is the acid, HCO3 minus plus H3O plus. So what will happen? From this proton will go here. So carbonic acid is formed, H2CO3 is formed, and water is a byproduct. So this H2CO3 is carbonic acid. This will decrease the blood pH to acidic side. So this is to prevent the blood from becoming acidic. Otherwise, blood pH will come down. To avoid that one, this bicarbonate takes that proton from this H3O plus and carbonic acid and water is formed. When a base enters the bloodstream, what will happen? OH minus ion. So at that time, this carbonic acid H2CO3 reacts with the OH minus because a base has entered inside your blood. So what you will get? You will get bicarbonate ion HCO3 minus plus water is formed as a byproduct. So this is to prevent the blood from becoming basic. So due to this, the concentration of H2CO3 carbonic acid and HCO3 minus is fluctuating. So here you see that at neutral pH both are equal, but when pH drops, their concentration fluctuating this side. If alkaline substance is added, their concentration will be fluctuating this side. So by fluctuating, it will maintain the blood pH without change, but here these two reactions will be shuttling. So here you can see the carbonic acid bicarbonate buffer system, how it works. Carbon dioxide combines with water and it will give you carbonic acid, a weak acid, H2CO3. So here only, inside this box only, carbonic acid bicarbonate buffer system functions. So this carbonic acid, when it is dissociated, you will get one proton from here, H plus HCO3 minus, it's a bicarbonate ion. So this bicarbonate ion can be kept as a reserve, bicarbonate reserve inside your blood. So where this bicarbonate goes as a reserve there, that may interact with the sodium ion present in your blood and you will get salt of this bicarbonate, sodium bicarbonate, NaHCO3. This will be preserved. Whenever you need this bicarbonate ion, the sodium is dissociated, the backward reaction. And so when you remove sodium, you will get HCO3 minus bicarbonate. That bicarbonate ion comes to the blood for maintaining the buffer system. So here you can see that sodium bicarbonate is there, that is dissociated into Na plus and HCO3 minus. This HCO3 minus come to the blood. It will react with proton. Suppose excess proton is produced in your blood. This proton is produced from these reactions. Fixed acids or organic acids, they will add proton to the blood. So when excess proton comes here, bicarbonate comes from this sodium bicarbonate, and now bicarbonate H plus ion interacts. It is going to become weak acid H2CO3 carbonic acid. Its concentration is increased here. When it is concentration increased here, automatically this H2CO3 is dissociated into carbon dioxide and water. Same this condition comes. So this is how, when excess proton enters inside our blood, bicarbonate carbonic acid is shuttling and it will act as a carbonic acid bicarbonate buffer system in your blood. So at the same time, what is going to happen in the other organs, like in lungs? This is the response to alkalosis. Suppose suddenly alkalosis increases in your blood, what is going to happen in the lungs? In the lungs, decreased respiratory rate comes, that elevates partial pressure of carbon dioxide, PCO2 increases in the lungs. When PCO2 increases in the lungs, more carbon dioxide will be there in the lungs. That more carbon dioxide what combines with water, H2O, and carbonic acid is formed. So this carbonic acid, it's a weak acid that will interact with proton, excess proton that is there in the blood. Blood is going to become acidic, but it will not allow. It will interact with the excess protein, excess proton, and it will remove that H plus ion from the blood because it will interact. So when it interacts, proton will be removed. How it will be removed? HCO3 combines with the proton and H2CO3 will be formed. If it is not necessary, this bicarbonate will be kept as a bicarbonate reserve for future use that combines with sodium ion, and sodium bicarbonate will be formed. That is kept as a reserve, NaHCO3. Whenever bicarbonate is needed, it will be dissociated, backward reaction. That HCO3 excess bicarbonate is formed, that will go to the kidney directly. Excess bicarbonate will be secreted through urine. Secretion of HCO3 minus through urine via the kidney cells. That whenever it becomes excess in the blood. When excess bicarbonate is secreted in the urine, at the same time in the blood, H plus ion is generated. That H plus ion comes to the blood. Similarly, proton may come from other buffer systems. They may also release H plus ion. That will also come to the blood. So this is called alkalosis condition of the blood. So where kidney and the lungs both are involved in it. And other buffer systems of the blood, hemoglobin buffer system. See that this is hemoglobin buffer system. First, this reaction is bicarbonate. That is carbon dioxide is there in the solution. That carbon dioxide combines with water, carbonic acid is formed, H2CO3. That will dissociate into H plus ion and HCO3 minus ion. This H plus ion may interact with this protein, that is hemoglobin, and it will interact with the proton, and oxyhemoglobin will be, or protonated hemoglobin, HB, is formed. So in that way, hemoglobin is going to act as a, in that way, this hemoglobin, HB, is going to act as a hemoglobin buffer system. Next one is plasma protein buffer system, protein buffer system. Here you can say this is the amino acid side chain of the R, the amino acid side chain of amino acid. When proton is removed, H plus ion is removed, you will get COO minus. When you add a proton, you will get COOH. This H1 is, this proton is removed, it will become ion. Or if you are adding proton, you will get, this OH is already there, you will get another proton added here. So in that way, this plasma protein is going to act as a buffer system. Next one is phosphate buffer system, HPO4 2 plus. That proton interacts with proton, you will get H2PO4 plus. This H2PO4 plus combines with OH minus ion, you get HPO4 2 plus and water. This is acting as a phosphate buffer system. So we'll come to this hemoglobin buffer system first. Hemoglobin, it's a very important protein present in your red blood corpus cells, RBC. This protein plays an important role more than other proteins as a buffer system, because this is a very important protein present in your RBC. Relatively high concentration in RBC. It is relatively rich histidine content, rich protein. pKa value 7.0. Its role is the important role of this hemoglobin in the RBC is transport of blood gases, that is transport of oxygen and carbon dioxide in the blood. So same hemoglobin acts as a buffer system also. So its role as a buffer system, it binds with carbon dioxide, binds and transport hydrogen and oxygen ion, participates in the chloride shift. It maintains blood pH as hemoglobin. It changes from two conditions, oxyhemoglobin. So this oxyhemoglobin curve, oxygen dissociation curve is there. It is PO2, it is millimeter mercury, 20, 40, a maximum 100 PO2. And this is hemoglobin saturation, percentage saturation of hemoglobin for oxygen. That is also 100% maximum here. So curve B is a normal curve, this curve B. It's a normal curve. A and C is abnormal. A means this curve. When you are going to get this shape in the A curve, when the conditions are like this, increased affinity for hemoglobin, so oxygen keep close. See that temperature decreases, the level of 2,3 diphosphoglycerol decreases, PCO2 decreases, pH increases, proton concentration decreases, pH increases. At that time you will get A curve. C curve is just opposite to A curve. pH decreases, proton concentration increases, PCO2 increases, 2,3 diphosphoglycerate level increases, temperature increases. So C is just opposite to A. B is the normal. B is the normal. Curve C is just opposite to curve A. Decreased affinity for hemoglobin, so oxygen released to tissues. So this oxygen dissociation curve changes between A and C when these conditions are changed. Otherwise, it will be normal. When blood pH is maintained normal, you will get only this B oxygen dissociation curve. That is the normal curve. Next one is protein buffer system. It originates from amino acids only. Already I have shown you. Albumin, it's a plasma protein. It's a primary protein due to high concentration in the plasma of the blood. This albumin is going to act as a very important protein buffer system. So buffer both, this albumin is going to buffer both hydrogen ions and carbon dioxide. So these proteins contain COOH group in the amino acids. Previous structure you have seen. COO minus groups. Like acetate ions, CH3CO minus acetate. It is also like acetate ion. It can act as a proton acceptor. So proteins also contain another group, amine group, NH3 plus groups, which like ammonium ion, NH4 plus. So they can act as donate protons, donors. If acid comes into the blood, excess proton comes into the blood, what will happen? Hydronium ions can be neutralized by this COO minus group. That is COO minus reacts with H3O plus and COOH will be formed, water will be formed. If base is added, what will happen? It can neutralize by the NH3 plus group that is present in the same amino acid. So NH3 plus combines with OH minus, NH2 is formed plus water is formed. This is how protein buffer system is functioning. So in this protein buffer system, proteins are made up of amino acids. Amino acids are playing a very important role in this protein buffer system. These amino acids, they have a central carbon with 4 groups off of it. First carbon is called carboxyl group, COOH. This is playing a very important role. Second one is an amino group, NH2. This is also playing important role in the buffer system. Third one is hydrogen atom. Fourth one is a side chain group, R group. So this amino group is attached to the amino acid central carbon C NH2. Neutral pH. When neutral pH comes, what will happen? The amino group is actually NH3 plus, always in the NH3 plus position rather than just in NH2 position. So in acidic medium, this becomes NH3 plus. In the basic medium, it will become NH2. So the carboxyl and amino groups are what enable they are enable proteins to act as a buffer systems. So this carboxyl group COOH, this is attached to the amino acid central carbon to this carbon. And this is also playing important role in the buffer system. So this carboxyl group consists of a double bond to one of the oxygens here in this one of the oxygen and a single bond to the hydroxyl group, this OH group. So at neutral pH, what will happen? The carboxyl ion, this COO minus ion, is present as COO minus instead of COOH. Acidic medium, it will become COOH. Basic medium, it will become COOH. So buffers are the compounds, or they are the mixtures of compounds that by their presence in a solution, they will resist changes in the pH, even 0.1, upon the addition of even small quantities or 1 drop of acid or alkali. That is their function, buffers. And you have another buffer system, phosphate buffer system, double salt buffers, it is called as. It is called as double salt buffer or phosphate buffer system. So besides the other two general types of buffers, that is acidic buffer or basic buffer, a third buffer appears to exist. This buffer system composed of two salts, that is this phosphate buffer system. In this, two salts are there. First salt is KH2PO4, monobasic potassium phosphate. Second one is K2HPO4, dibasic potassium phosphate. These two are combining and it forms a phosphate buffer system. So in this phosphate buffer system, main elements of the phosphate buffer system are these two only, H2PO4 minus and HPO4 2 minus. Phosphoric acid changes pretty quickly into dihydrogen phosphate, that is H2PO4 minus position, first one. This dihydrogen phosphate is an efficient buffer. So this phosphate buffer system in the extracellular fluid, that is ECF, fluid outside the cell is called extracellular fluid. It is very, very, very low compared to the bicarbonate buffer that is there in the blood. Its buffering power is less. It is very less. It is very less compared to the bicarbonate buffer. It is very less compared to bicarbonate buffer. So it has its importance in the renal tubules, that is in the kidney, renal tubules of the kidneys for two important reasons. So here you can see the phosphate buffer system, pKa of phosphate buffer 2.1, pH is 6.9, 12.8 maximum. H3PO4 that can be converted to H2PO4 minus plus proton, or that can be converted to HPO4 2 minus plus proton, or PO4 3 minus plus proton. So this phosphate buffer system, these two salt HPO4 2 minus and H2PO4 minus plays a very, very, very important role in plasma and erythrocytes of the blood. So H2PO4 minus interacts with water, H3O plus is formed and HPO4 2 minus is formed. Salt. Any acid reacts with monohydrogen phosphate, this one, monohydrogen phosphate, to form dihydrogen phosphate, this H2PO4, dihydrogen phosphate. So dihydrogen phosphate H2PO4 combines with water, you will get monohydrogen phosphate HPO4 2 minus plus H3O plus. So the base is neutralized by dihydrogen phosphate. So here you have dihydrogen phosphate H2PO4 minus. When OH comes, HPO4 2 minus is formed, H3O plus is formed. You will get monohydrogen phosphate, this one.